we got the screens on, because that would have been a heartache if we wouldn't have had them on. Everybody got their PowerPoint handouts? On your lecture packets, have that out, and open to page 8. We're going to be going page 8 through 11. Actually, through 12. Lecture packets. Okay. Everybody pay attention. This is how you tackle difficult, big topics. Because this is not the only one, and in the future, it will be bigger and more difficult. Okay? What you do is you don't get intimidated. That doesn't help anybody. So don't be. Rather, take a step back, see the big picture, and remember the context. Are we clear? Mm-hmm. Then you take this big, heavy issue and you split it up into sequential steps. Each one you could do. It's just a lot of them. Learn it in sequence. And by the time you're done, you'll actually understand the whole thing. And what you understand, you don't have to memorize. Are we clear? What are you supposed to do when you have a big, intense topic?
Break it down.
Yeah, I'm gonna go over it again, because this is a life lesson you guys really should get straight. Do not be intimidated. That doesn't help anybody. That's first. Don't be intimidated. That doesn't help anybody. Second, take a step back, see the big picture of what this is for, and learn the context. The context makes all the steps make sense. You don't have the context, it's just stuff, it's just content. You guys get that? Mm-hmm. Then break it up into smaller sequential steps. Each one you could do. Why you want to get intimidated is because it is so many of them. Don't be intimidated. Take a step back and see what this is for. Remember the context, because that's what makes all the detailed content make sense. If you don't remember the context, this is just stuff that you're going to try to memorize like this is anatomy, and that won't work. You learned how to do this in physiology, and you might actually pass pharmacology when you're in the program, which is the first class that weeds people out in the nursing program, which is way harder and less organized, I understand, than this. Are we clear? Are you guys ready to do this? Because we're going to do this all today. Here is what the whole big picture, the reason why we're studying this. We are studying how cells are going to use these 3 links to generate ATP, which is the only energy cells use. They don't use glucose, they use glucose to make ATP, and that's the only energy cells use. That's the context. You guys got what the context is? How do cells use the 3 nutrient links to make ATP? Now what we're going to do is we're going to start with this framework first. And this is 3 sequential steps. Once I have this built first, then I can add the other ones to it. But I'm going to do monosaccharides first. Now this is, we talked about this is catabolism. Taking starch, say, from a hamburger bun, and you're going to break it down into individual monosaccharides. Monosaccharides is what your body absorbs and your digestive system goes into your blood. That's what the cells get a hold of. from food. Started out as food. Everybody got it? That's why these three are called nutrients. So far, so good? You're going to break down chains into links, and this link has 6 carbons in it. H2O for every carbon. We're going to even continue the catabolism so that we're going to go to 6 CO2s. It's all catabolism. We're breaking down a 6 carbon molecule into 6 CO2s. That's catabolism. But this catabolism is what your GI tract does. This is what your cells do. That's the topic. Does everybody have the context? Remember it, otherwise you're going to get lost in about 10 minutes. Remember this context. First, it's how our cells use the 3 nutrient links to make ATP, the only type of energy cells use. I'm going to start it with monosaccharides, then I'm going to build in the other 2. This is all about catabolism that releases energy. First, your digestive system does this. That's once you have the links, your cells do this. This is about what's going on in the cell. The topic is cellular respiration. It is what's going on in the cell. Now, last week, we built some of the preliminary principles. We talked about electron carriers, which are also RNA nucleotides. They're dinucleotides. We talked about decarboxylation reactions. All that is important background information. I don't have time to go over today. In fact, I'm going to have to start picking up right now. Are we ready to go? Do you remember that stuff we talked about last Wednesday? That needs to be in your head. I can't keep reviewing it. You have to be on it. Okay, this slide was one of the last slides from last week. 3 sequential steps. First is glycolysis, Krebs cycle, and then the ETS, what we call oxidative phosphorylation. Wherever you have carbons, that is substrate. So if you make ATP where there's carbons, that is substrate level phosphorylation. What happens in step 3? There are no carbons left. That's what we call oxidative phosphorylation. Does that ring a bell from last week? Yes. All right. First step is glycolysis. Go ahead and fill in the blanks. Glycolysis literally means glucose splitting. This happens in the cytoplasm. Now it's a whole sequence of steps, but the bottom line is a 6-carbon glucose is going to be split into 2 3-carbon pyruvates. That's up on the screen in the blue box. You do need to know that. You do not need to know the biochemistry of all the individual steps with all the individual chemical formulas. It's big picture stuff. I'm asking you, this is not a biochemistry class, and this is way, way dumbed down. Now, one of the things in chemistry you got to know is that you might hear it called pyruvic acid. or you might hear it called pyruvate. It, for our purposes, is the same thing, hence the equal sign. This means it has the H on the carboxyl group. This means the H fell off, which is why it turned the solution acidic. You guys remember the carboxyl group? It either have the H or not have the H. This has the H, this doesn't have the H. For our purposes, it's the same thing. Okay, it's going to go through a whole series of reactions. One of the things that you got to do is you got to put 2 ATPs in. You get 4 ATPs out. So this is 2 net ATPs. Because this is where the carbons are. This is substrate level phosphorylation. What it says here at the bottom bullet. You also have to put 2, I should do this in green maybe. Does it help when I color code things?
Yeah.
It's a pain in the ass though, you know that, don't you? 2 NAD, and it's sometimes referred to as NAD plus, don't worry about it. And you're gonna get 2 NADH plus H. Okay. You can just call it NADH. But remember there's a pair, so there's actually 2 hydrogens on it. This is in the cytoplasm. This does not require oxygen. Your cells are going to do this whether there's oxygen or not. I've just covered all the bullets on this slide. Read it. Tell me if I'm wrong. That first bullet is why you have to have 2 ATPs go in. So let's run this animation. You can actually see what biochemistry students have to go through. Here's the 2 ATPs dropping off of high energy phosphate group. It becomes different types of sugars. It's actually called fructose. Here's the second one. Those are the 2 ATPs in. Already it splits into 2-3 carbon molecules. It's just not pyruvate yet. Here's the 2 NADHs picking up high energy electrons. Here's 2 ATPs coming out. And here's 3 and 4 going out. 2 ATPs in, 4 out. When I was in San Jose State, we had to know not only the names of all the intermediate steps, but also all the chemical formulas and the enzymes. So if you tell me this is hard, I've seen hard. This is not hard. You can get the big picture things. This is what I had to learn when I was at San Jose State. You want me to hold you to that? That's from your textbook. No, okay, I won't. I'm holding you to this, the big picture summaries. Everybody got it? Also, you must be able to visually recognize glycolysis at least to this level. You should be able to recognize 1 6 carbon going to 2 3 carbons as glycolysis. Any questions? Okay. I do have to have a couple slides on anaerobic respiration because this first step could be with or without oxygen. If there is no oxygen, we're going to do this 1 reaction anaerobic respiration. It's just 1 step. Go ahead and fill in those blanks while I do this. This is also happening in the cytoplasm. Now, I'm not gonna do this for both halves of this. But know that if there's without oxygen, it's gonna do this one step, anaerobic respiration. The enzyme here in red is lactic acid dehydrogenase, not LA Dodgers. And that one reaction is going to change pyruvic acid into lactic acid, or we can call it lactate. Same thing as this, lactate, pyruvate, same thing. Everybody got it? You will see it both ways in textbooks and online, so I want you to be aware of pyruvic acid, pyruvate, lactic acid, lactate. The bummer here is that to make lactic acid, you gotta utilize these high energy electrons you got from glycolysis. So this is all burned right here in green, it's gone. And the only thing you get when you do anaerobic respiration is the 2 net ATPs up there in glucose. That's all you get, that's the third diamond. 2 lactates, because both of them are doing it, so this will be both. And 2 net ATPs from glycolysis. And ADHs are wiped off. Yeast cells have a different enzyme. They don't have lactic acid dehydrogenase, and when they do anaerobic respiration, they produce ethanol, which is why anaerobic respiration is also referred to as fermentation. Those are synonyms. Don't you wish you had that enzyme that yeast cells have? All you gotta do is run around the track, and it's a party. You guys get what I'm saying?
Yes.
If you had the same enzymes as yeast cells, all you have to do is feel the burn, but you're not gonna feel the burn, because you're not gonna make lactic acid, you're gonna get drunk. Because that enzyme results in ethanol. In fact, when you make fermented drinks, whether it's beer or whiskey, it's yeast cells making the ethanol because of their enzyme. But you gotta make sure there's no oxygen because it's anaerobic respiration. Everybody got it? So far so good? Is it overwhelming so far? There's a lot more to it. I'm just telling you what you need to know. Okay? Again, both of them would do this. If there's no oxygen, both halves will do the same thing, but I don't have enough whiteboard, so I'm just gonna split it. Are we clear? Any questions? Now, most cells will do anaerobic respiration only in emergencies, like you're stuck underwater too long, you can't breathe. Red blood cells, and this is the last diamond, and skeletal muscles do this routinely. In fact, let's talk about skeletal muscle doing this routinely. Again, this is from your textbook, this is all you gotta know. Here's the name of the enzyme, lactic acid dehydrogenase, or just lactate dehydrogenase. And again, the NADHs are gonna get burned, so whatever you got from glycolysis is gone. And the only thing you get are 2 lactates and 2 net ATPs, that's it. Red blood cells definitely are doing this. They don't have much metabolism anyway, they're pretty inactive. But definitely in venous blood, there's low oxygen in venous blood, so red blood cells do this in venous blood. Skeletal muscle will do this routinely because their demand for ATP can jump 100-fold between rest and exercise. So that's why we have what is called the Cori cycle. Go ahead and fill in the blanks, you can go over this. Okay. Because skeletal muscle does this routinely, your liver is going to form this circle. And it's going to minimize you feeling the burn. You're still going to feel a burn, but it's going to allow you to do anaerobic respiration longer. For example, even if you were a top world-class athlete, you can only do anaerobic respiration for 60 to 90 seconds. That is a top world-class athlete. For most of the students in this room, at about 20 seconds of anaerobic respiration, you're crying like a baby, because it's burning. Now, the only reason why world-class athletes can stretch it out a minute to 90 seconds is because of the Cori cycle. What it's doing is it's taking the lactate that's released from skeletal muscle, turning it back to pyruvate. Lactate and pyruvate are not carbohydrates. They don't have a water for every carbon. But the liver is going to put two of these pyruvates back together and form glucose. This is new glucose from non-carbohydrate sources. What is that called? Gluconeogenesis. If you didn't know that, it's highlighted here, and it's on the figure here. All you gotta do is read it. New glucose from non-carbohydrate sources. Pyruvate is not a carbohydrate. It's going to take that glucose, spit it back out in the blood as free glucose. Once it gets into the muscle, it's gotta have a high energy phosphate group on it, and that's what glucose 6P means. It's got a high energy phosphate group on the number 6 carbon. That's what glucose 6P means. Glucose travels in your blood without phosphates, but glucose does nothing in your cells unless it's got a high energy phosphate on it. That's why it has to have that put back on in here. Then it can go through it again, turn it back into lactate. Liver's gonna get the lactate, turn it back into pyruvate, back into glucose with gluconeogenesis, put it out in the blood as free glucose. Then the muscle can use it again, and that's why a high world-class athlete can do anaerobic respiration for that long. It's because of the Cori cycle. If it wasn't for the liver doing the Cori cycle, that world-class athlete would be crying like a baby after 20 seconds too. Does this make sense? There would not be in track and field the 800 meter. Those are world-class athletes, but they're sprinting 2 laps around the track. There wouldn't even be that event without the Cori cycle. Do we understand what the Cori cycle is for? Again, you have to be a world-class athlete to have this actually make a difference in you. If you're a couch potato, 20 seconds, you're trying like a baby anyway. Any questions? All right, get the essay sheet out for lecture exam 1. I just covered essays 2 and 3. 2 is about glycolysis, but it's the bottom line things. Where does it occur? What goes in? What comes out? Again, that's all the if I ask that question on lecture exam 1, just give me the content. It's just got to be in complete sentences, as it says on the top of the sheet. Cori cycle. That's essay number 3. These words are copied and pasted from the PowerPoint slide. We actually have covered most of these. We covered all full back page. We covered 1, 2, and 3. I'm going to cover the rest of them today. Are we ready? We've just done 1 of 3 steps. That is 1 of 3 steps. Let's go to the second step. Now for the second and third steps, you must have oxygen. If you don't have oxygen, you only do this. That's it. This is also where we have to involve the mitochondrion, and hopefully that was covered in your anatomy class. I don't know, maybe I should do it over here. This is my band, by the way. Our first gig is this Saturday at Harry's in Pismo. My bandmates make me do that. You have to know the anatomy of the mitochondrion for this to make sense. You have 2 membranes. You have the outer membrane. You got the inner membrane. The single ring DNA is somewhere in the matrix. And then you have 2 spaces. Inside the inner membrane is the matrix. In between the membranes is the intermembrane space. You need to know that if you're going to understand the steps 2 and 3. If you look on page 9 in your lecture packet, it's showing these same things. These little finger projections of the inner membrane is called the crista. Little fingers of inner membrane. That's called the crista. That's on that page 9. Everybody see that? Mm-hmm. So the matrix is all the way on the bottom right. Cytoplasm is all the way on the top left. You can see the 2 different membranes of the mitochondrion, outer membrane, inner membrane. Inside the inner membrane is the matrix. Between the 2 membranes is called the intermembrane space. Okay? So this is where we're at when we're talking about the Krebs cycle. We're actually in the matrix. You can actually see that. Top left on page 9, glycolysis is happening just out in the cytoplasm, but the Krebs cycle is in the center of the mitochondrion in the matrix. The third step, the electron transport system, also referred to as the electron transport chain, is on the inner membrane. So, do this in orange. Krebs happens here. His name was Krebs, by the way, so you don't need the apostrophe. And on the inner mitochondrial membrane is the electron transport chain. So this is step 2. This is step 3. And this here is step 1, glycolysis. The 3 steps. I'll get an orange. Does that help? Mm-hmm. So far, so good? Are we ready to go on to the second step? Stop me now, because it'll be much more difficult to backtrack later. So we have 2 pyruvates. Again, with oxygen, it's going to be both of these. I'm going to go through this. but the whiteboard isn't big enough. First, we have to have a linking step. And I actually showed the picture of the linking step before when I was talking about decarboxylation. In week 2, when I covered decarboxylation, it was the same picture. 3 carbons, CO2 comes off, decarboxylation becomes 2 carbons. This is going to be an acetyl group. In organic chemistry, 2 carbons is acetyl. And when you have this catabolism, that is, energy is going to be out, like it says there in purple, that energy out is going to go onto a high energy electrons on NADH plus H. It's going to put a coenzyme on the 2 carbons, and it's coenzyme A, so we call this acetyl-CoA. This is what is referred to as the linking step, or intermediate step. Some textbooks call it prep step. Since oxygen is required, we call this aerobic cellular respiration. So it's steps 1, 2, and 3 for aerobic cellular respiration. For anaerobic respiration, it's only step 1.
Mm-hmm. Yeah, I know.
And it's the acetyl-CoA that actually enters the Krebs cycle. Just remember this last bullet, both are doing it. If you have oxygen, both are doing it. So you have 2 Krebs cycles going per glucose. Both of them are going to go through it. I'm just splitting it up because the whiteboard is only so big. Everybody understand? Are you still tracking? Don't be bleeding on the tracks, man. Stay on the train. Because it's pulling out of station 1 going into station 2. This is the prep step. 3 carbons goes to 2 carbons. CO2 comes off, but we get NADH, but remember there's 2 of them, because both of them are doing this. Okay. I'm not going to ask you to know all the intermediates, only the first reactants and the first product. And the same thing goes here with acid and 8. Oxaloacetic acid is also oxaloacetate. Same thing. Everybody get that? Do I have to say that anymore? Does everybody get that, or do I have to keep saying it? Does everybody get it, that it's the same, or do I have to keep saying it? Because whatever I don't finish today, you're going to have to do on your own. Okay. The reason why you have to know these names is because some textbooks will not call it the Krebs cycle. They will call it the citric acid cycle. And it's for the first product of this cycle. Everybody got it? 2 carbon acetyl group from acetyl-CoA is going to hook onto oxaloacetic acid, becoming citric acid, 6 carbons. 2 plus 4 equals 6. Everybody got it? Again, you don't need to know citrate, isocitrate, alpha-ketoglutarate, succinate, you don't have to know all that. You just have to know these, but you do have to know the net products. So let's just go over this. It is a cycle. It's happening in the mitochondrial matrix. Let's see, I need green. Looking at big picture thing here, you don't have to have all the details of this. So all the ATPs, you know, is coming from ADP plus P, right? It's putting the last phosphate group on ADP. So I'm just gonna listen like this. What kind of phosphorylation is this? What is it?
Yeah, substrate.
You see C's around here? You see carbons? This is substrate level phosphorylation. Wherever you see ATP made, where there are carbons, that is substrate level phosphorylation. All right. One more. Here's the only place we find FAD. Again, you should be able to visually recognize the Krebs cycle. You don't have to fill it out, you just gotta visually recognize it. You should be able to visually recognize 6C going to 2, 3Cs. You should be able to see this Krebs cycle. Any questions? So per every cycle, we're gonna get one ATP, which is a substrate level phosphorylation, because that's where the carbons are. It's the last one, by the way. You're gonna get 3 NADHs and 1 FADH2. Does everybody see that? Do you see it in the figure? Do you see it on the whiteboard? Do you see it in the bullets? All right, then somebody answer me this, and this is why on test questions, including the TEAS test, you must read the question and all the choices with ruthless accuracy, because this is not a typo. Each one of these guys takes up a pair of high energy electrons. Everybody got it? 1, 2, 3 pairs, 4 pairs, this is not a typo, 5 pairs of electrons. Somebody explain that to me.
So double what you got,
Huh?
double what you got, what you're getting, this is gonna go through again.
Well, there is 2 cycles per glucose, but I'm just talking about 1 cycle, half of it. This is just 1 cycle, which is half of it, because there are 2 cycles per glucose. This side here, if you had oxygen, would be just like that. You have 2 per glucose if you have oxygen. Either both are not gonna do that because you don't have oxygen, and both will look like this. But if you do have oxygen, both of the sides are gonna look like that. Does everybody get what I did on the whiteboard? You're not gonna make me write this out twice, right? Just think of all the things you're gonna have to learn on your own. If I just spend the time to write them both out. Why does it say 5? Read it with ruthless accuracy.
Is it because of the 2 hydrogens on the FADH?
It's either gonna be written like that or written like that, so it's the same
Same
thing.
thing.
1 pair, 2 pair, 3 pair, 4 pair. Why does it say 5?
Because it's coming from the original pyruvate.
The original pyruvate includes the linking step, 5. Pyruvate's up here, people. Everybody got it? It's all on the whiteboard. Any questions? That is essay number 4. Any questions? Three is happening on the inner mitochondrial membrane, and that's the ETS. That's the third step. Again, you must have oxygen. You'll see why here in a second. Aren't you glad you don't have to memorize all this? I kid you not, I had to write out the structures. I had to write these out for isocitrate. I had to memorize this whole dang thing. I've seen hard. This is not hard. This is what you got. Which is kind of like what's on the whiteboard, isn't it? Also, okay, we started out with 6 carbons, right? Right?
Yes.
I'm only doing half. This 3 carbon side, 1, 2, 3. All the carbons are gone. Because if this is also on this side, because you have oxygen, both sides are doing it, all 6 are gone. Does everybody see that by the time, in fact, I would write that, all the carbons are gone after this step. All 6 carbons are gone after this step. These 3, and this side would be just like it if you had oxygen. These 3, all 6, are now CO2. We've just done this in the first 2 steps. All 6. Substrates are gone. No more substrates left, but all the ATP that you see here and here, remember there's 2 of them, so this would be 2 ATP. All of that was substrate level phosphorylation. But now the substrates are gone. They're all now 6 CO2s. Does everybody see that? Are you tracking? Everybody look at me. Absolutely nowhere on this planet will you see cellular respiration explain this straightforward and this simply. Nowhere. So you better grab onto each word that comes out of my mouth, because any other option you have is going to be less clear or more complicated. This is it. This is the bottom, and it's taken me 15 years to do it, to bring it down to the most basic bottom level. This is it. There's no way to cover this any more basic. This is it. Okay? ETS, it's on the inner mitochondrial membrane, as it says over there. Here now, instead of carbons, we have all the high energy electrons on these electron carriers. And you can see it looks like the fluid mosaic model of cell membranes here, don't it? You got protein boulders floating in a sea of phospholipid bilayers. Everybody see that? It's just an inner mitochondrial membrane. Looks just like it. Except these boulders are all electron carriers. Each of these protein complexes, each of these boulders, will take the high energy electrons from those green guys and pass it down to the next boulder. So it's just going on. I'm not going to draw all the tails and the heads. But it's going to take off the high energy electrons off of NADH. And turn it back into NAD. What happens to the NAD? It goes back to the first two steps. Then it's going to take that high energy electron. I should get another color. And I guess it's a pair of electrons. Just going to pass it on down the line. Okay? So these guys come in a reduced state. They drop off the electrons in the first boulder. This becomes reduced, this becomes oxidized. Then this reduced boulder drops it off to the next boulder. This becomes oxidized, this becomes reduced. Then this one passes to the next one. This becomes oxidized, this becomes reduced. Each one of these boulders is just like the electron carriers. They are just alternating between reduced, the high energy state, and oxidized, the low energy state. They're just passing it down the line. It's right here in yellow. Boom, 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 boom. So each of these boulders first becomes an electron acceptor, becomes reduced, then it passes it on, becomes an electron donor. Each time the electron is passed, energy is released. I'm going to explain that later. This is why you need oxygen. This is why oxygen is required. Because by the very end, and remember these electrons are not free electrons, they're going to go onto half an oxygen. And two of those is going to become H2O. Water is the, oxygen is the final electron acceptor, becoming water. And you can see it right here. Everybody got it? Now we're going to make a lot of ATP, but there are no carbons left. This is not substrate level because there's no carbons left. This is oxidative phosphorylation. Now the last two essays here on 5 and 6 are going to be answered right now. This is basically the summary of the ETS, of what's going on. There's actually a little side boulder here. electrons from NADH and FADH2 do not drop off on the same boulder. So take a look, this is somewhat similar to what I have on the whiteboard, this figure here. NADH drops off the 2 electrons at the very first boulder. FADH2 drops it off 1 step down. Theoretically, the electrons from NADH will yield 3 ATPs, but because FADH2 skips the first step, it only generates 2 ATPs. So if you look at this figure, if these electrons are going from left to right, eventually hooking onto oxygen becoming water, correct? Following, are you guys following that? If the NADH electrons get dropped here, you're gonna get all 3 ATPs. Going from left to right. Are you seeing that in the figure? But since FADH2 skips the first boulder, its electron starts here. So it's only gonna get 2. Does everybody see that? Okay? These are theoretical yields. And on the bottom of page 10 is the ATP scorecard. Let me get your mind at ease about
Is
this.
this paper?
10 more minutes to do this. I'm going with 36. Your newest editions of the textbook, your textbook also used to do 36. It went to 38. These would be ideal theoretical yields. Are you following me? The problem is that no cell does that. There are costs, transport costs of getting these molecules in and out of the mitochondria. In the real world, no cell probably even makes 30. Probably the top end for any cell for 1 glucose is about 29. And the rest of them are lower than that. Those are the real yields, actual yields. This is what I'm talking about right now here on the bottom. Well, if no cell does even 30, why the hell am I gonna go to 38? So I'm just sticking with 36. You just gotta know, in the real world, it's not this many. This is a theoretical yield. Are we clear? And we're gonna go with 2 because there's 2 cycles per glucose here, and 32 here in the electron transport chain. There's your 36. 2, 2, and 32. If you look on the figures that we covered in week 2 PowerPoint, that's what it says, 2, 2, and 32. Okay? No cell does that. Well, if they don't do 36, they sure as hell don't do 38. So I'm not going with the new editions of the textbook. I'm sticking with 36 as an ideal yield. But this is the simplified form. This is vertical instead of horizontal. And again, NADH is going to get all 3 of these. FADH2 electrons skips the first one, so it's going to only get 2. Everybody see the difference? Everybody see the difference? Everybody see the difference? Don't stop engaging. Your programs want me to tell you, stay on this. Is it hard? Yes. But you've got a lot more harder subjects coming up. And if you just veg now, you've got to think about doing something else. The programs will not want you if you do not hang on. You hanging on? Okay. Now this is where we're going to talk about the chemiosmotic theory. And this is actually essay number 5. The context of the chemiosmotic theory, since contexts are important to understanding, is that this is a more precise explanation of what's recurring, what's actually occurring in step 3. This is a more precise explanation of step 3. It's developed in the early 60s. The electron transport system that I showed you there on the whiteboard and in this figure that's on this slide is an oversimplified inaccuracy. It's a lie. Because ATPs are not synthesized at the site of the electron transport reactions, the redox reactions. ATP are not made there. That's a lie. The chemiosmotic theory is a more precise explanation of how this is actually happening. I said early on, and you might have missed this because there's just so much going on here. But on this slide, actually it was on the very first slide. Yeah, that was actually mentioned. 3 membrane protein complexes, 3 boulders, and then highlighted proton pumps. Did you guys even notice that before?
Mm-mm.
Okay, these are also referred to as proton pumps. Because the reality is, as the electrons are being passed from one boulder to the next, it is actually pumping hydrogen ion into the intermembrane space. It is pumping hydrogen ion from the matrix to the intermembrane space. This is causing a great concentration gradient, but since this is protons that are positively charged, this concentration gradient yields a pH gradient. This is the H of pH, as well as a voltage gradient, what we call membrane potential. Because there's a bunch of positive ions in the intermembrane space, and it takes a ton of energy to make and maintain those gradients. That is what is actually happening. Then a little bit further on, so this is the intermembrane space here. This is the matrix down here. On that inner mitochondrial membrane, in addition to the 3 proton pump boulders, there is this protein channel called respiratory assemblies. And so since you have all these free protons up here in the intermembrane space, because all 3 of these guys are doing that, the only way the H pluses can go downstream, what kind of transport is this? When ions go through a protein channel. This is facilitated diffusion. And these protons are flying through it because there's so many protons in the intermembrane space. It's flying down back to the matrix through these respiratory assemblies. Attached on the matrix side of the respiratory assembly is the globular enzyme ATP synthase. So this is now, I'm jumping down to essay number 6. as the protons are flying through the respiratory assemblies, it is spinning ATP synthase like a pinwheel in a rotary fashion. And every rotation around, 3 ATP are being made by oxidative phosphorylation. This is one of the most efficient energy transfers in biological systems. This right here. Everybody see that animation? Every complete revolution around, 3 ATPs are being made. Because it is so efficient, this is where we get most of the ATPs out of the 36. So, step 1, we get 2 net ATPs. Step 2, we get 2 ATPs. Because remember, there's 2 cycles per glucose. So one on this one on that one. 2 both of these, so 4 ATPs come from substrate level phosphorylation. This is going to get us 32 ATPs by oxidative phosphorylation. These are substrate level phosphorylations. Why? This is where the carbons are. The 32 come from oxidative phosphorylation, but you only get these 32 and these 2 if you have oxygen. If you don't have oxygen, you only get these 2. That's it. So what is more efficient for getting your cells ATP? Aerobic respiration. You get 36. Without oxygen, you only get 2. Are we clear? This is sequential thinking that I just built right here in orange. How are we going to go from a glucose, which is monosaccharides, through this 3 steps to get us 36 ATPs? That's the framework. Now, I got a couple minutes before I plug in these 2. Is there any questions about this framework? Is that a ton of stuff? It's going to get way harder. This is not a ton of stuff. But it's okay to feel like it's a ton of stuff. But when you're looking for a teacher, you want one who is clear and consistent and organized. That one will make it easy. Any questions? Yeah, this is where your textbook says 34, that's why they say 38. No, let's just stick with 32. There's some animations, you can look up things on YouTube. You are responsible for basically what I'm giving you, which is a small percentage of the 3 steps. And I just covered all the rest of the essays. No more essays. Most of them were from today though, huh? 1, 2, 3, 4, well, half of them was from today's lecture. All right, now that we have this framework, this is why this is sequential thinking. Everybody look at me. This is why sequential thinking is so important. The 3 sequential steps of this framework, starting from here, that's got to be established, that's there. Now we can add into it the other 2. But this is still there. Are we clear? Mm-hmm. This is the topic of intermediary metabolism. If you take a biochemistry class, this is like 2/3 to 3/4 of the whole class is just on this. Now, look at this figure, it's not that difficult, everybody look at me. It's not that difficult. What we just established is what is in this figure in black. Everybody see that? That's why I do that first. The black is established. Now I'm just gonna add in the red and the blue. Everybody see that? Okay. Let's do the red first. So this is lipids with fatty acids. You do have to recognize for lecture exam 1 the sequence of breaking down fats. Let's see if I can do this. I'm working my ass off here. All right. A triglyceride is a glycerol with 3 fatty acids. And I'm not gonna draw all the C's, but I'm gonna draw it out like this. Because the fatty acids are long chains. Remember the phospholipid bilayer has 2 of these tails? Triglyceride has 3. Glycerol, and that's the reason why I put it here, is a real short 3-carbon molecule. And on this end is the carboxyl group, OH, double bond to oxygen, OH, double bond to oxygen, OH, double bond to oxygen. So this is a triglyceride. You must be able to visually recognize this for lab exam 1, 2. Don't you wish you had chemistry now?
Yes.
Are you pissed off at this college for not requiring it as a prerequisite? You should. In fact, you should go up high up in the administration to complain about it. Especially since we're the only college that doesn't require it. Glycerol plus 3 fatty acids. Everybody got that? You must recognize this reaction as lipolysis. And the reason why we're able to get this OH group over here is because 3 waters went in there and split it. What kind of reaction is that? There's other ones similar to that. What kind of reaction is lipolysis? Three waters went in to split it.
Or hydrolysis.
Hydrolysis reaction. This is catabolism. One big molecule gets split into 4 smaller ones. One big molecule, this is 1, is now 4, glycerol and 3 fatty acids. Everybody see how that's catabolism? In fact, it's right here. A triglyceride is a lipid. It's going to go through catabolism to get you fatty acids and glycerol. It's all on the whiteboard. Everybody see that? That happens first. And that's up here. Lipolysis happens first. Now, we're going to take one of these fatty acids. And even though I didn't draw all the carbons out, let's say there's 16 carbons in that. The carboxyl group on the end. Everybody see that? It's going to go through several reactions. It's not just one. And you're going to make it into 2 carbon segments. This is beta oxidation. All these arrows, it's multiple reactions, beta oxidation. Okay, so it's first lipolysis up at the top, then it's beta oxidation. And you're gonna get 2 carbon acetyl groups that will now get put in here. They're gonna put a coenzyme A on it, boom, it's gonna enter 8 Krebs cycles. Because there are 8 acetyl groups out of 16 carbons. Because each acetyl group is 2 carbons. Everybody got that? Well, if you go 8 Krebs cycles, look how many NADHs with an FADH2, and you're also going to get substrate level phosphorylation. One of these fatty acid, one of these lines is going to get you 108 ATPs. And a triglyceride has 3 of them. Triglyceride is going to get you over 300. And that is why, up there highlighted, why lipids, triglycerides, are a major source of stored energy. A triglyceride can get you over 300 ATPs. Okay? This also illustrates the central role of acetyl-CoA. Not just for fats. There's a lot of things that'll go through acetyl-CoA. But here's the triglyceride. So this green arrow is lipolysis. Happens first. This green arrow is beta oxidation. And then that should be about 4 arrows. And then that's going to get you a ton of ATPs. But acetyl-CoA is a central molecule of intermediary metabolism. Your old 4th edition of the textbook, they got rid of these figures, and I really thought that was stupid because it did it really clear. The center was the thing that we started with. We established that first. Now we know where triglycerides go. Fatty acids are going to go into acetyl-CoA, but also the glycerol can actually be made into glucose, gluconeogenesis. Proteins. Go ahead and fill in the blanks. I said last week you got to recognize the NCC of amino acids, right? Well, we only need the carbons, so you got to take the N off. And that's the process called oxidative deamination. You're taking the N off. Now you just got carbons that can be plugged in to the framework that we already built. By the way, you must, I already told you, you gotta visually recognize amino acids, the NCC, as well as the peptide bond between NCC links. Okay? So again, once you take off the N, the nitrogen off of oxidative deamination, now what do you do with it? Well, you put it on ammonia. You just add another hydrogen to the end, and that's ammonia. You could put that amino group on others and make other amino acids. That's called transamination. Or you can convert it into urea in the liver, which has 2, look over here on the bottom right, has 2 amino groups, that's urea. You can get rid of 2 of the N's in urea, but just, it's highlighted there because I want you to know this. It's your liver that makes urea. Your kidneys just filter it out and put it in your pee. But the kidneys do not make urea. It is your liver that makes urea. Your kidneys just filter it out and puts it in your pee. The CCs, the carbon chains, can go in multiple places. along the framework we just built. It can go into pyruvate, it can go into acetyl-CoA, and it can go into various intermediaries of the Krebs cycle. Each one of these Cs is what's referred to as a keto acid. Those are the acids of the Krebs cycle. They're all called keto acids. And once it's in there, as one of those keto acids, it's contributing to the Krebs cycle. So you're gonna get ATPs from that. So again, you take the N off, you can put it on urea, or you can make other amino acids. You guys know we have 20 amino acids in all our proteins, there's only 20 links. And we can actually make 12 of them. There's only 8 we can't make that we must eat. Those are the essential amino acids. But the 12 that we can make is from transamination. Okay? Let's here, we covered where glycerol fatty acids go. This is where amino acids go. They go into pyruvate, acetyl-CoA, or the keto acids of the Krebs cycles. Do you see why we had to establish the middle first? We do that first, then we can do the 2 things on the sides. So all 3 links your cells will use to make ATP. We established this first, then we added these other 2. Again, there's a lot more to it than that, but you are gonna be held to what I gave you. That's how you know how in depth you have to be. Any questions? Even though I started late.